Welcome to a concise introduction to ADCs, or antibody-drug conjugates, which marry the targeting precision of antibodies with the potency of cytotoxic drugs. Think of them as guided cancer bullets that deliver toxic payloads directly to tumor cells while sparing normal tissue. And in this presentation, we'll walk through how they work, why they matter, and where innovation is heading. The evolution of ADCs reads like a story of technical learning. Early attempts in the 1980s struggled with unstable linkers and weak payloads, leading to poor efficacy and safety. Lessons learned on linker chemistry and ultra-potent payloads led to the first approvals around 2000. And today, the field has exploded to over 300 candidates across oncology and beyond, driven by improved stability and broader indications. ADCs function through a four-step sequence that must all work. First, the antibody binds the target and the complex is internalized. Second, it traffics to lysosomes. Third, the linker is cleaved and the payload released. And finally, the drug diffuses to its intracellular target, inducing apoptosis. Successful delivery requires coordination at each step to convert binding into meaningful tumor cell death. Choosing a linker is a balancing act between cleavable and non-cleavable strategies. Cleavable linkers respond to lysosomal pH or proteases and can enable bystander killing, while non-cleavable linkers rely on antibody catabolism for controlled release and improved plasma stability. Selection hinges on tumor biology, bystander needs, and manufacturing feasibility to optimize therapeutic index for each indication. Auristatins and maytansinoids are microtubule targeting payloads known for picomolar potency, which makes them extremely effective, but also risky. Their hydrophobic nature can drive bystander killing, useful in heterogeneous tumors, yet premature release raises neurotoxicity concerns. This demands meticulous linker tuning, so you get targeted delivery without excessive off-target toxicity. DNA-damaging agents and topoisomerase inhibitors offer a complementary payload class. Calychiamicins and duocarmycins cleave DNA strands, while topoisomerase I inhibitors like SN38 block relegation, both favoring quiescent and proliferating tumor cells. These agents require stable circulation and efficient lysosomal liberation to achieve potent apoptosis, making linker stability and intracellular release strategy essential design considerations. Clinical validation came with agents like T-DM1 and brentuximab vedotin, which demonstrated real survival benefits and transformed standards of care in HER2-positive breast cancer and Hodgkin lymphoma, respectively. Those successes validated the ADC platform, catalyzed regulatory pathways, and attracted significant investment, accelerating broader development across solid tumors and hematologic malignancies. Development is moving beyond established targets like HER2 and CD30 toward novel antigens such as TROP2, HER3, c-Met, FRA, and CD79B. Tumor biology, expression patterns, internalization rates, and normal tissue distribution now guides linker and payload choices, enabling a precision oncology approach where target characteristics dictate ADC engineering and therapeutic index. Resistance to ADCs arises through multiple mechanisms. Antigen downregulation limits binding, impaired internalization blocks, payload delivery, efflux pumps, export drugs, and target mutations can render payloads ineffective. Strategic combinations aim to overcome these barriers by pairing ADCs with checkpoint inhibitors, deploying alternate payload classes, or using bispecific formats to restore activity and extend durable clinical responses. Off-target toxicities remain a clinical challenge when payloads are released outside tumors, manifesting as neutropenia, ocular events, and peripheral neuropathy. Active mitigation strategies include optimized dosing regimens, prophylactic eye care, and tumor-activated or conditional linkers designed to release payloads only in the tumor microenvironment, all intended to widen the therapeutic window and improve patient quality of life. Dual antigen binding. ADCs offer enhanced selectivity by requiring simultaneous engagement of two targets, which reduces on-target off-tumor toxicity and promotes co-internalization with improved lysosomal trafficking. Early candidates pair antigens like HER2 with LAMP3 or CD19 with CD22, and this approach promises better precision, lower systemic toxicity, and efficacy in heterogeneous or antigen-low tumors. The future of ADCs centers on personalization and global accessibility, integrating gene expression signatures, real-time imaging tracers, and AI-driven dosing to tailor therapy to individual patients. At the same time, continuous manufacturing and improved linker payload chemistry aim to lower costs and scale production, so ADCs can become mainstream, accessible precision medicines across diverse healthcare systems.